Good morning and welcome to the panel on policy and regulation. My name is Peter Phillips and I'm replacing Dame Ann Glover, who was unable to attend because of a family bereavement. So today we have an esteemed panel of both people who think deeply about regulation and policy, but also have been practitioners. They've been at the coalface of making and managing and using the policy and regulatory systems that we've created in Canada and more globally. Let me make a couple of introductory comments, and then I'm going to invite each of them to make an opening address. Then there's going to be an opportunity for you to speak. We are being taped, so I will do my Vanna routine. I'll have a microphone, which I'll come around and ask you to speak into the microphone so that when we actually have the end product, we can hear your question and not just the answer. So let me sort of set the stage. Regulation is often perceived as something governments do. I think this panel will explore that a bit in the sense that regulation goes well beyond the government. It's really a multi-leveled, multi-party process that's about ensuring safe and efficacious technologies, not just in the food space, but across our economies, enter the market and get used by citizens. In that sense, it's a highly structured, and some would argue over-structured system. And that system has a variety of levels that people are interested in discussing. At one level, there's the nation state. We used to have a system in the food space where countries would invent a technology, they would assess it, they would approve it, and then they would export both the technology and the products thereof. And pretty much the rest of the world would through mutual recognition or other processes, accept the decisions of the inventors and the producers. We've moved a long way from that. We now have not just the 30 mega adopters of GMOs and other advanced technologies regulating, but every importing country has an interest in regulating some or all of the questions about that. On top of that, we've layered what I call a spaghetti soup, an alphabet soup of international organizations. The WTO and its sub-agreements on sanitary and phytosanitary, trade barriers, or non-trade or technical barriers to trade and investment and intellectual property systems, the Codex and OIE systems, and IPPC systems in the standard setting space, and then a whole bunch of almost sui generis systems. The OECD, some of the people on the panel have been part of their negotiations and discussions, but all kinds of bilateral and multilateral trade processes, TPT, TTIP, CETA, the Quads. We have a large body of people trying to structure the system. And meanwhile, beyond that, firms have their own internal regulatory processes. NGOs are heavily involved in trying to structure the norms and questions we would ask in this space. And scholars and scientists, whether they're in firms, governments, or in the academy, are all interested in contributing evidence or input to the policy process. So we've ended up with quite a complex system that's having significant impacts that are becoming codified. And so regulation is an important element to consider when we talk about new technology. Stuart the other day mentioned the Phillips-McDougall study, which put a gross estimate of about $136 million on the development and first stage adoption of new technology in a global setting. About 27% of that funding, that $136 million, is for regulation, and a large portion of the time involved, and time is money in the commercial channel, is involved in developing evidence for or assessing the evidence that goes into the regulatory structures. And so we have a longer, more expensive, and more uncertain regulatory system, which is having effects. Greg Graff and colleagues from Colorado State have estimated that of 560 proof of concepts in the bioscience space that they were able to identify a number of years back, only 383 got to trials. So somebody made a judgment that even though they were proof of concept, that they didn't warrant further investment. 47 went to advanced trials. Only 14 were presented to regulators for decisions, and only 5 got through. So less than 1% got through, and of those 5, only 2 actually survived in the market. So the 2 that survived are carrying in many ways, the costs of the other 558 candidates. So the regulatory system is a sieve that's very tight, very sharp, very focused, and that reduces in the eyes of industry and in the eyes of the investors, whether it's Gates or the Government of Canada or USDA or the European FP system, it reduces the ROI, in particular because of some opportunity costs, and we get less investment. Some of us have estimated that it could be as much as 2 major traits a year are foregone because of the investment chill because of delays across the system. So I'm pleased to introduce a panel that is partly culpable in creating the system and is tasked with, in many cases, managing the system. I'll introduce them sequentially, and then we'll bring them up to say some few words. We have Phil Macdonald, who's been with the Canadian Food Inspection Agency from the beginning, almost from the beginning. He came out of the NRC in Ag Canada, where he applied. He worked on cell signaling and the science that he now regulates. He, as with our last speaker, or our last on the panel, were involved in helping to set up the Canadian system of plants with novel traits, which is a somewhat novel but quite robust process that we have in Canada for regulating these. Second on the panel is Ruth Onyango, who's the editor-in-chief of the African Journal of Food, Agriculture, Nutrition and Development. She has a PhD in food science and nutrition and has taught at universities in Nairobi and elsewhere. She was a member of the Kenya's parliament and was the lead author of the Kenya Biosafety Bill, which has subsequently led to the first field trials of biotech crops in Africa. So she brings a very applied, real-world experience in the markets that we're interested in moving these technologies to. Third, we have Greg Jokas, the Canadian Consul General, just back from Bangalore, India. He has 30 years of experience in trade policy and diplomacy, with an emphasis on agricultural processes in Canada and abroad. He's recently returned from India and has in the past been a Director of Trade Policy at Agriculture and Agri-Food Canada. So he is involved in the policy space that structures the systems we have to deal with. And finally, we have Stephen Yarrow, who those of us who have been around this process a long time still think of as one of the faces we know in the Canadian regulatory system. As a PhD scientist, he moved into the Canadian system and has been involved for the better part of 30 years in working in the plant biotech policy and regulatory space. He spent 19 years with the Canadian government and was part of the team that developed the PNT process in Canada. I think everybody at this table has been part of the Cartegena Protocol and some of the other bilateral, multilateral processes. And currently, Stephen is Vice President of Plant Biotech at CropLife Canada. Now, I've invited each panelist to speak for about 10 minutes. Some of them have slides, some of them don't. And then I'll do my routine of coming out and inviting you to comment, question, probe, challenge the comments, and we'll try and wrap up as close as possible to the deadline. Phil.
Thanks very much for that warm introduction, Peter. God, those lights are bright. I feel like the regulator's inquisition. So, as a regulator, I came over prepared already. I wasn't clear on the task, so I'm the one with lots and lots of slides. But what I'm going to try and do, maybe, let's see if I can get this to work. Which one is the green button? Okay. So one of the things I'm going to talk about a little bit is why the Canadian system is as good as it is, and how we kind of heard a lot of the messages that you've heard. Stuart talked about how a broken system works. I'm going to try to show you how a not broken system works, and how we're trying to make some positive changes to embrace the kinds of technologies that you've heard about here today. Particularly, there's really quite a moving speech that I talked from this morning, and actually my graduate work was actually on photosynthesis and related to herbicide tolerance, and it was really really quite moving for me to hear how much work and how much progress is now being done. So Canada started off on the right foot, and I think, you know, what you hear a lot of today in the current government is evidence-based decision-making and going to science, and that's really where the Canadian system started. And I think one of the things that Peter didn't mention is that biotech is a little bit unique in the fact that really the discussion has started in the public space. There was the Asilomar conference in the 70s where people really said, you know, we have to really think about how we're going to regulate this technology. So the industry kind of stood back and said, whoa, you know, we better get some advice and some input. So that already kind of put the discussion on the public space, and it's a place that I think people still feel they own if you think about last night's talk and the angry stakeholder knocking signs over. So it's a place where we try and tread that place between innovation and between making sure that the Canadian public feels that they're protected. So in that light, right at the very outset in Canada, in 1993, a regulatory framework was crafted. And the regulatory framework really has stood the test of time. And it talks about what are our perceptions for how we're going to regulate the technology? How do we feel about that? What do we really, where are we going to position that in kind of our whole policy space? So what we've come back to, and I think the key issues here is that really this is about regulating those products, protecting the environment, but it's also allowing innovation to proceed. And that's a clear message from the policy environment. And it also talks about using existing legislation. So that puts those things firmly in the context of the things that we know. So that's kind of where we started right from the very beginning. This is about making sure that we protect Canadians, but allowing innovation to proceed, and also putting it into the context of the kinds of products that we're already familiar with. So unlike a lot of other jurisdictions, there is no Gene Act or biotechnology agency. So we use existing acts and legislation. So right from the very beginning, the world said, you know, back in the 80s, that, and this is kind of the message that you heard last night as well, there are no unique risks from the use of molecular techniques of genetic engineering, which are extensions or refinements of earlier, less precise, less predictable techniques. The same physical and biological laws govern the response of organisms modified by modern molecular and cellular methods and those produced by classical methods. So this is a statement from the NRC in 1989. So we took that to heart. We took that really to heart. That's what our own Canadian scientific community was telling us when we went out and consulted. And it came back, and that's why in Canada, unique in the world, we don't talk about GMOs. We don't have to talk about GMOs. We talk about plants with novel traits. And a plant with novel trait can be created by any kind of methods. It's agnostic to the methodologies that you use. So it's not necessarily something which has been produced using recombinant DNA technologies. So take a minute to digest that. So as we move into an environment where people talk about, you know, the CRISPR-Cas9 kinds of technologies and genome editing, and we look at, you know, cisgenics and site-directed mutagenesis, and what does this all really mean in the world, and people are bending themselves out of shape because about how are they going to regulate these kinds of things. Back in 1993, we'd already been there. People had already decided that we really needed to be forthright. So now we have a continuum of products. We have truly, unique in the world, a product-based system. So the science, we got the science right at the outset. So in the Canadian context, a GMO can be a P&T, but a P&T is not always a GMO. So not every plant with a novel trait is a GMO. It's bimodal. You have to be new to the species and potentially pose a risk to the environment. And I'm going to get to near the end. A GMO is not always a P&T. And that's truly a unique concept for Canadians. So having said all that, people still worry about GMOs. I mean, actually, it's really kind of ironic that what's pictured here is kudzu, which is not a GMO, but it is an invasive plant. And we spend a whole lot more time worrying about genetic engineering and crop plants that we know and putting a single gene in than we do about bringing whole things in like this, whole genomes, whole species, invasive plants. We have safety assessments to try and cover off the concerns that people have around the outside of the technology. So you can probably imagine, and this really kind of describes the bimodal system here in Canada. At the top, this would be a field trial. So we keep all our potential hazards. We don't know whether there's crocodiles out there or not. So, and as long as we keep those away from any kind of exposure, then there should be no risk. And that should actually be a multiplication sign. But once the crocodiles are out of the cage, then all bets are off. So what we're really hoping is there are no crocodiles. And we're basing this very much on our safety assessments rely on our knowledge. And if you go back to think about the regulatory context, we talk about this being in the context of things that we know. And that's very much what we talk about. We have biology documents that describe the species, and that's our baseline for doing those kinds of assessments. And we rely on substantial equivalence. These are our key concepts. So you can well imagine, we know about canola, we know about corn, we know about how those things grow in the environment. So when when we start thinking about substantial equivalence, you think about, well, if I know about canola, what happens if I put another gene in there? And in fact, when I was with the Royal Society when they did the review of our biotech regulations, and someone from the Royal Society put the question to me, how do you determine the effects on biodiversity of Roundup Ready canola? And I said, well, you have to stand back and think, what are the effects on biodiversity of growing 8 million acres of canola? It's pretty substantial. So that's our context, that's substantial equivalence. If a canola gene's got 30,000 genes in it, so if we put that additional gene in, what's it like in that context when we start thinking about those, or what are the effects and the interactions in the environment? So we rely on a familiar counterpart, our unmodified species. We use the concept of substantial equivalence to inform what's acceptable risk. And this has been very effective for us for the most part. And really, we have a pretty good record, well over 100 crops authorized now for release for food, feed, and the environment. And this is our assessment criteria. We call this our mighty 5 pillars. These are usually in one form or another. This is basically how people design their conceptual models, the kinds of things. What do we care? What is it? What do we care about, and what are we trying to protect? But having said that, as you can well see, people don't always embrace new technologies. And again, we've had to be very cautious as we proceeded. And that's been probably one of the hindrances in the regulatory process, is we haven't really been a learning system. We've tried to be more of a learning system, and we probably haven't evolved as quickly because we're also mindful of the fact that we're dealing with our own stakeholder community. And there have been unexpected outcomes as we've moved along through here. And there's no question that things can always step up to create problems for you. So one of the things, for example, and this was during my early days, and I think Stephen was my boss at the time, or maybe it was Morven who was my boss, and this was, somebody made the surprising discovery that if you fed poisons to insects that was toxic to them, that they died. So if you think back to my model with the crocodiles and the duck, that really, what we're really talking about here is that the crocodile, the exposure, that's the important piece. And this has nothing about exposure. So we already knew that. And if you go back and go back and look at that paper and look at the dose and response curve, and if there's any toxicologists out there, it doesn't work. Then, of course, there's our friend Dr. Sarah Laney, who has spent a long time, and there's the big discussions about glyphosate now in the European Union and the issues around the outside of glyphosate. And I'd like to note that this paper, actually, they were forced to retract it. So what's happened out of this? We talked a little bit yesterday about what this has really meant. I think that was our talk from last night. So if you look here, what we're really talking about now with the adoption of herbicide-tolerant canola in Canada, there's been a true shift towards zero-till farming systems. A market shift. That's a real change from environmental sustainability. So we're mindful for those things as we move forward. So how are we trying to learn? We're in a new environment, like I already said, where we have a variety of product challenges. One of the biggest challenges that we're facing right now is genomics. If you really think about it, if you think back to the original days of the Human Genome Project, when people were really talking about spending 10 years trying to sequence the human genome, this is work now done in weeks. We're looking at a whole range of technologies that are pushing us along with genomics, not just a huge volume of information. So right now, we already have products in the pipeline using these new genome editing technologies. And in fact, I got a phone call from the Austrian government. They're worried because one of their canola products, which is already, they have something which is very similar in the pipeline already, which is done with actually chemical mutagenesis. So here's one using site-directed mutagenesis, and the Austrian government is bending themselves out of shape trying to figure out what to do with that. So they said, well, we have a risk assessment. You can have a look. You can figure out how to conduct those risk assessments because they haven't done any of their own yet. So as we move forward, nothing's really changed. As we fast forward 20-some years, people are still saying the same thing, that genetic engineering itself doesn't pose unique risks. It's the kind of products that you produce, which really endorses what we said right from the very beginning. So here's the most recent thing, I'm sure, the National Academy of Science report, which Henry Miller just slammed because it was too cautious. And Henry Miller, unfortunately, also invoked genomics, which is the bête noire for regulators, because, as I'm fond of saying, I have a lot of responsibilities in the genomics sphere. Genomics is a wonderful technology because you can generate enormous amounts of information. Genomics is a horrible technology because you can generate enormous amounts of information. So as a regulator, all that really does is provide a whole lot more questions for you and not a whole lot more certainty. So we're looking to improve the comparative approach. We're looking to try and figure out how best to deal with that thing. So our first step actually, and I know I don't have enough time, Peter, but I'm going to invite you to have a look. This is really unique in Canada. This is a piece of peer-reviewed scientific advice that we have provided, generic scientific advice on plants with novel traits. And this is really how do we think about new transformations. So if you know you have a gene and you put it into a crop and you take it and you have another transformation event using the same gene, what do you do with that? What does that really mean? So for us, in that context, we said, you know, really, those risk assessments still apply. And that sounds like a small thing. Does that really matter? But if you're an apple producer or a potato producer, and all of a sudden when you move into the regulatory process, and every time you do that transformation for another variety, you don't have to go through a whole new regulatory process, that's huge. It also telegraphs to the community where we're going with the science, how our science is growing and evolving, and how we're thinking about the changes. So I think this is critical, and this is all three regulatory bodies that are responsible for the oversight. That's the environment, the food, and the feed side. So this is kind of a critical step for us, and I would invite you to go have a look. It's certainly created a lot of discussion and a lot of interest in the rest of the world. And I think especially as people are looking at genomics and the amount of information that comes out of there, it's very timely that we have the genomic science where we started some years back. Now genomics is backing up what we thought in the first place. So to finish, Peter, the ERA for genetic engineered crops, it's a relatively new science. We haven't really been at this very long. And we've started at the highest level of precaution. There's no question about that. I mean, guilty as charged. We came to a place where we really didn't know what we were doing. We were all trying to learn. I mean, the day I started, nobody had done any kind of a review on these at all. They've been sitting there, a big box of files sitting there, and people, well, what do we do? So we really did have to learn. I mean, you don't come in there learning how to be a risk assessor. And common practice and often our regulations tend to bind us. I mean, we're creatures of habit, and you don't become a risk assessor because you're a swashbuckling risk taker. You don't go into government because you're really looking to be an entrepreneur. So the kind of personalities that get attracted to this, you tend to be pretty cautious, and we like to stick to our routines. And it's not really surprising that this is really the way the regulatory system is. And we also have to be conscious of our stakeholder community, that Canadians are all not at the same level of risk tolerance either. So our regulatory framework has guided us to make sure that our oversights and accept some risks in our outcomes. So we've still made decisions. We still made decisions in an imperfect world. And the regulator's mantra is need to know versus nice to know. And that's where we always start off when you're learning. Is this a question you're asking because you need to know the answer? Or is this because your scientific curiosity needs to be satisfied? So genomics is going to be a significant challenge for risk assessors for the reasons that I've already touched on. But I think one of the things that we do more and more now is look for more structured approaches to figure out how to attack problems. What are pathways to harm? What is that need to know versus nice to know? These are the things that really help us structure our risk assessments. And we've got a lot of experience with plant breeding, and familiarity is an important concept for us. It's a place we go back to all the time. And I'd like to finish off by saying that, Canada, we have far from a perfect regulatory system, but I think we really do have one of the best. We started off on the right foot. I think we've continued on the right foot, and we're continuing to try and change and evolve and learn in an environment where the technology really doesn't stop. Anyway, I think I'll stop there, Peter.